Our next speaker is Dr. Mary Hardy. She's a physician. She is going to be speaking on the concept of safety and its measurement and its reporting. Dr. Hardy is board certified in internal medicine, a specialist in botanical and integrative medicine. She has really a lot of extensive experience in evaluating the evidence-based for the efficacy and safety of complementary and integrative medicine practices. She's currently affiliated with the George Washington University Medical School's Integrative Medicine program. This is Dr. Hardy's first presentation at our Practicum, so please give her a hearty and warm welcome.
Hi, everybody. I first would like to thank the organizers for inviting me. It's nice to be here. It's nice to see what a practicum is. It's certainly been eye-opening and very practical for me as well. So I'm going to talk to you about safety, and luckily this program was very well designed because I think that there's been a number of complementary presentations that touch on each other, so that I was afraid I would have to give you the entire context, which had been half of yesterday and everything that happened this morning. So happily, I don't have to do that in 30 minutes. Ultimately, every assessment of whether or not to use a supplement, drug, whatever, comes down to a balance of risk versus benefit. Now, I'm not going to talk about benefit today. Joanna's talked a lot about patient values. The benefits already been touched on as well. But this issue of risk is, I think, a critical component, especially when the benefit is either not very pronounced, not thought to be very pronounced. But it's important to also know that risk varies depending on which chair you're sitting in. So risk for a regulator will be different for risk for a patient who may feel more compelled about the disease entity they're trying to treat than perhaps a risk as assessed by a health agency. And ultimately, most safe use of these things comes down to an appropriate interaction between issues related to the patient and issues related to product quality. Yesterday, Adam talked a lot about all the issues around standardization, evaluation of product, and product quality in the issue of looking at dietary supplements, but especially for herbal products, is a critical issue. And so before you do any other assessment, you want to make sure that what you think you're looking at is what you're actually looking at. Now, patients, and I call them patients, most other people call them consumers, and therein is part of the problem. Many products will be used without supervision, without appropriateness for use. So, for example, someone who has hypertension, who's taking a product that may affect blood pressure adversely, again, not always the best a mix of patient and product. The product itself may be made well and without inherent risk, let's say, but in that patient, it's an inappropriate choice. So we talked about what other medical issues they may have, but also stage of life. Metabolism varies depending on your age and your other medications, et cetera. In looking at the adverse event reports for Ephedra, which we're going to talk about at the end of this presentation, it made a huge difference what state the person was in to begin the adverse event report. So being dehydrated, actively in exertion, et cetera. External environment. It was a very different matter taking this product on a cool fall day versus in a hot sauna, for example. And we're going to talk a lot subsequently. Dr. Gurley's going to talk about interactions and supplements, and that allows me to skip that point. There also can be some racial or ethnic variation, both on some of the SNPs and the way things are metabolized in patients, but also into other activities patient populations may or may not use, as Joanna had already told us. Now, if you're going to look broadly at quality issues, this is the list. And again, we're going to talk a little bit about some of these. Normal but toxic doesn't happen that often. Now, when we say intentionally added, that's usually drugs, not toxins. Contaminants, and there'll be times when the manufacturer made a mistake, when they just put in 10 or 20 or sometimes a higher amount of any substance. And the reason why we want to kind of look at the at the sort of the whole picture of this is because the remedies are going to vary. If it's a mistake in manufacturing, fine. If it's a contamination, that's one thing. Inappropriate patient population, inappropriate labeling, that's what the FTC is for, et cetera. So you kind of want to start to take globally the things I'm telling you and start to triage them based on what would I do with this information, how would I make an intervention that would provide a greater safety environment for my patient. Our beloved Norm Farnsworth, who's not with us anymore, has said, and I believe this is actually true, that if what's on the label of an herbal medicine and a dietary supplement is what's in the container, toxicity rarely is encountered. And so again, that critical first step, getting what you think is in the container in the container. Adverse events can come from a variety of different sources. We talked a little about adulteration, inherent toxicity. These substances can have a pharmacological effect, which is minor. It may be a direct action. It may be mediated through the CYP, the liver enzyme detoxification system. It can interact with other drugs or dietary supplements. Patients can misuse any product. They can double, triple the dose. And then after all that's said and done, people just do weird things, which is medically known as idiosyncratic reactions. So contamination, you're going to notice if you're following along with my slides, that was my first cut at a really low dose of slides, a small number of slides, but I've gone further than that. So you're going to have more slides in your handout than I'm going to talk about now. Those are there mostly for another example, the same thing I'm talking about, additional papers to bring to your attention, or definitions. There's a whole bunch of definitions that I didn't figure you needed me to read to you. All right, so contaminations. This is looking at a study that looked at, reviewed the literature and looked at a number of reported adulterations. Now, there are unintended adulterations, and some of these things, seaweed, for example, can have heavy metals. They're just present in the water. And calcium supplements, especially those that are made from oyster shells, can have heavy metals in them. Again, environmental contamination working into the basic product source. This perhaps is a bit more problematic just because it's evil-minded. You have economic adulteration where you take something that's well known and then you substitute a cheaper or different compound for that. And some of it is a different herb, some of it's putting in a compound that will make the marker compound look higher than it really is. We had a whole spate of reports in, especially on the West Coast, Chinese patent medicines were contaminated with drugs. If it was a patent medicine used for pain, there might be an actual pain medication, either over the counter or a usually prescribed medication adulterating. The highest risk products for intentional adulteration are erectile dysfunction products, which have a lot of sildenafil analogs in them, performance enhancement, which will often contain steroids or other banned substance, and weight loss products. These are the highest risk products for contamination. Misidentification can often be an issue. And there's a couple of examples in your handout. This is one that I thought was particularly interesting. I was serving on the Natural Health Products of the Canada advisory panel when these 6 adverse events were reported. On the basis of these adverse events, a question came up whether or not to label all black cohosh products as potentially being hepatotoxic. Well, going back to my first principles, I asked that we would assure that what we thought was in the bottle was actually on the bottle. It turned out it wasn't. It was, instead of an Actaeus species, it was Chinese black cohosh, Actaeus fetida instead. So these did not have the product in it, so therefore labeling all black cohosh products, which are supposed to be the American version of black cohosh, Semicifuga Actaea racemosa, as having a problem when in fact there were constituents that were more commonly found, natural but toxic constituents that were normally found in this herb that were not present in the material that actually got in the bottle, because it was cheaper to buy a Semicifuga, a black cohosh from China than to use the actual correct species. So that's just a quick dash through some of the things that you've heard a little bit about, looking at it specifically from a safety issue. Now, if we're going to make an evaluation, we have to get our information from someplace. So where can we find useful information? So the first place I like to start is to talk to other people who've had experience using a particular herb. They often will know astonishing amounts about the herbs they're looking at, and that includes working with native traditional healers in several different places across the globe, and most closely in Peru in the Amazon. And this patient, this provider, this healer, this herbal expert would know things about the plants that were just astonishing to me. Then you want to have a sense of, have there been pharmacologic or mechanism of action studies? That might give you a reason to suspect that something might be the cause of an adverse event or might potentially lead to an adverse event. What toxic constituents might be present? Look at pharmacovigilance reports. People publish things in conference abstracts, don't complete, observational data. But the biggest chunk of what we look at is adverse event reports, and that gets a lot of weight put on it, and it gets a lot of interest and attention. There are adverse events reported in clinical trials, controlled, randomized, case-controlled trials, but also just, out of the blue, something really bad happened, and this is what the person was taking, sort of, post hoc reports. There's a very small amount of pooled data, but mostly there's not enough data to begin with, so there's rarely enough data to actually do a pooled analysis for risk or adverse effects. So, and we're going to go through these topics in more detail because depending on where you're getting your information from, each of them will have particular risks and benefits, limitations associated with them. The first thing to recommend to your attention is that people who are a committed herbal user, committed dietary supplement user, are going to be very unlikely to report. Reports in general are in the single digits, and if it's a committed user who doesn't want to make trouble for the product that they like, then they're not going to report it. And they're less likely to report an herb than a drug if they're a committed user. So most reports that are made are not severe. Most of the ones that get the most attention are in fact very severe, but they're quite rare. So if you just go to the medical literature, because that's your sort of second impulse, just go look it up. Someone must have written about this. It's actually quite difficult to find this information. It's not housed very neatly under a single mesh term. If you use adverse or adverse effects, complications, poisonings, to get to a fairly high degree of certainty that you've identified most of the events that you're going to want to look at, you have to put together multiple terms. And even at that, you're going to have to winnow away a lot of other stuff. So just finding these events in the literature is not that easy. So let's go back to our historical reporter. Why can't you just talk to someone who's had lots of experience using an herb? Well, if someone's taking an herb, whatever the law says that you can use an herb or supplement for, people mainly still don't maintain a healthy mental status. They hear depression when they think about St. John's wort, for example. So the question that was asked earlier about, efficacy, and you can't use it for a label claim, but you have to test for something. That's a very apt comment, and patients tend to make that translation. So many of the things that we're being treated for here in a Western first world society are nothing like the traditional societies. They didn't really have atherosclerotic vascular disease in most of these countries and most of these societies because people died earlier of something completely different, or they had a completely different diet or a completely different exercise regimen. So, but there are times when between traditional and Western there can be tight concordance. I did an analysis looking at herbs of use for Ayurvedic herbs of use for diabetes. And in fact, the concordance between the diagnosis in Ayurveda, Madhu Meda, and what we call diabetes was quite close. But you had an easily measurable outcome that you could do even in a low technological society. So diabetes mellitus means honey. The sugar gets sweet in a diabetic. So there was a way to make that diagnosis and follow that patient in a low technology system. So in general, this kind of safety data is best for acute toxicity. If you give someone a poison, you know pretty quickly, right? But for chronic toxicity or unusual toxicity, not very, very useful. Then we take a traditional preparation and we substitute an herb, or we refine how it's taken. So chaparral is a bush you'd see in the west of the United States. It's got a very oily, resinous texture because it's in a desert. So if you just drink that at a tea, you're not going to extract a lot of the key ingredients because they don't dissolve very well. But take that same thing and either take whole herb and put it in a capsule or extract it with alcohol, and you've now significantly changed the traditional method of preparation, and in fact, you actually significantly changed the toxicity of that. So you have to be aware of that because everyone wants to borrow traditional data for the safety of their intervention, and that's fine as long as it's an equivalent analysis. When you look at clinical trials, a lot of clinical trials don't collect adverse event data, and they don't collect it very well. They'll do some testing to look for unexpected adverse events, routine labs, et cetera. But there's a section always someplace, 2 or 3 sentences that the paragraph says the product was well tolerated. So let's assume that your trial was successfully randomized, not a benign assumption, and that you've got 2 balanced groups. Then if something does show up, more often in the verum group than in the placebo group, you can reasonably say, yep, that can be attributed, causality can be attributed. But it's, again, not usually thought of very carefully when clinical trials are run. And part of my pitch today is those of you who are doing clinical trials, pay a little extra time here, and you can derive great benefits for the rest of us. The most expensive part of a trial is finding the subjects, randomizing them, and following them. So if we can just have you start to think about adding in a more robust safety assessment, it doesn't cost that much more, you can start to really build this progressive database as was talked about in the last presentation. Now, so clinical studies, randomized controlled trials, these are our gold standard. Why can't we just do this for everything? Why do we have to have any other kind of study design? Why do we even care about these adverse event reports? Well, it's because most clinical trials are very poorly powered to find unlikely events. So a clinical trial will find a common event that occurs frequently, that occurs shortly within the initiation of a medication. For most trials are pretty short. But most trials aren't maybe long enough to find a long-term effect. But the biggest problem is that if you want to guarantee that you're going to find something that happens once in 1,000 times, which is not that rare, you need to enroll 3 times as many subjects. So the idea of finding an uncommon or even a not-so-common adverse event in a clinical trial is largely going to not happen. The numbers are against you. You're, again, the specialty population that was discussed, underreporting by observers, how you get your reports, and then there can be event bias, how you're questioned, how you're asked about this, etc. So this is one of these long slides that tells you all the ways in which we generally don't do things very well. But specifically for safety studies, the drug was generally well tolerated. Well, what does that mean? Some people had a little bit of stomach upset. Only one person really threw up a lot. What? So not being specific, not providing the actual data, not providing the actual numbers. In general, just not being specific enough. Now, there are 2 kinds of ways to elicit these adverse event reports. The one is the researchers will just wait for someone to spontaneously report something to them. Spontaneous reporting is generally very underreported because anything can make you feel a little wonky for a couple of days. It's difficult to assess. On the other hand, if someone specifically starts to ask you, did you have this, did you have that, did you have that, that can predispose you, can bias you towards having those side effects. I was included in a hepatitis vaccine trial as an intern, and I came back to the study examiner after 2 weeks and said, God, my arm's been killing me, and I felt like I had the flu the whole time. And the guy looked at me and goes, yeah, you are in the placebo group, so. So, okay, you can really suggest things that will happen to people if you aren't careful about the way in which you solicit. But I do think some combination of passive and active should really be included. And this just sets you towards reminding you that there are consort guides for reporting, that we want to be sure that we can generalize, that what we do is valid, etc. So again, just pay a little bit more attention to this aspect of your trial work. Now, looking at adverse event case reports, which is where the biggest bulk of this analysis comes from, and where most people get all bent out of shape about stuff. First of all, if you look at the adverse event reports, you're immediately crippled, because most of the, remember I told you, I think the first move you have to make is what's in the bottle what you thought it was? And most of the data sets for looking at these large dietary supplements, a large number of adverse dietary supplement events, I looked at 80 event reports, and they actually did not have an adequate description. One had an adequate enough description that I could even be sure of what they thought what was in the bottle was in the bottle. Rarely, if ever, is the material ever recovered, collected, and tested. So this is in an individual, and it becomes a very big problem and as you go forward. Pharmacovigilance is just a name for sort of post-marketing surveillance. It's usually a voluntary process, very underreported. It cannot prove causation under any circumstances. Even if you have 10,000 cases, it's technically not a proof of causation, but it will raise your index of suspicion and identify something that you might want to pay more attention to. Causality can be proven maybe in a case cohort trial, maybe in a randomized trial, with the kind of limitations I already told you about. So causality, at the end of the day, becomes the name of the game. Someone reports something terrible that happens. What changes that from a sorry for you to, we better really think about this some more kind of an event? And that's, is what they told you was the adverse cause, is it the adverse cause? And how do you go about assigning that causality? In your set of definitions, there are two scales that will tell you sort of the formal way that this causality is assigned. But I want to walk you back through some of this stuff. And again, think about the kind of data you're looking at so that you have a realistic expectation for what you can and cannot get out of the kind of data you're looking at. Randomized controlled trial, that's the point of the whole thing. Things that happen in one arm don't happen in the other arm. If it's well randomized, should be able to be attributed with all the caveats. Cohort, maybe. Case series, nope. The only kind of adverse event that points towards real causality for the material you're interested in is something called a de-challenge or re-challenge event. So let's say I tell you I got sick to my stomach from taking a dietary supplement. That's my report. So you tell me, take away that dietary supplement, go about your business, tell us what happens. Well, my stomach upset, resolved. Now that's kind of interesting, still not causality. So let's say, because you're this kind of a person, you really like to just poke your finger in things to make sure that what you think is what you think. You decide, I'm going to start taking that again just to see if it really did cause my nausea. Sure enough, you take it again, and the same symptom comes back. All right, that's the tightest kind of causality you can get out of an adverse event report. Now, immediately you can see there's certain problems. If death is your adverse outcome, I can't re-challenge you. If a severe heart attack or stroke or seizure is your adverse event, I'm not going to want to re-challenge you. So automatically, for the more serious outcomes in adverse event reporting from case reports, you're going to be severely limited in then being able to assign causality. And there's a bunch of other different kinds of studies which kind of amount to sort of some post-marketing, some other kind of case control studies, retrospective historical controls, all that kind of stuff. You can't attribute causality. Now, if you are going to try to attribute causality and you want to see how tightly you can get this linkage to link up together, you're going to want to know how severe this is. Obviously, if it's minor and self-limiting, goes away after a little while, whether you stop or don't stop your supplement or your dietary supplement or your herb, It's important that it's reversible. That's pretty helpful, because if you've had an adverse event and it's a completed adverse event, then there's less way to intervene. You want to make sure that the pharmacology or the exposure linkage with the adverse event is appropriate and seems logical based on what you know about your substance. In the Ephedra adverse event reports, and I read hundreds and hundreds of them, people would say, I took this and it caused me to have a stroke. Well, when did you take it? It was a week before my event. That's this as a direct causal agent, no additional doses in between, that doesn't match with pharmacology. But if we have someone who took something and the onset of the side effect or the onset of the acute event was appropriate for the pharmacology and pharmacokinetics, that makes this a lot more likely to be true. The one that really gets everybody into a lot of trouble is this one, the absence of alternative explanations. Now, this usually requires someone with medical training to make this assessment. And it's classic in abnormal liver function tests, liver toxicity, adverse event reports. Pretty much everything, including the party last night, can cause your liver function enzymes to go up mildly, okay? A couple of drinks can really do that by the next day of measurement. So, Tylenol, there's lots of drugs that can affect the liver. So trying to rule out other events that could cause liver abnormalities is quite challenging, and it takes very thorough reporting to really show that there is no other alternative explanation. The more explanations you eliminate, the tighter your relationship gets to be between the intervention that you think caused the problem and the observed problem. So keep that in mind as well. Mostly when we start to look at this stuff, just even getting the information, we don't often know everything. We don't often have all the information. We may not know what to ask because we're not clear what the mechanism of action of a product is, et cetera. And there's a lot of variability, both from person to person, and so we can't extrapolate very easily. Well, I saw one person that had this happen to them, therefore everybody who's like this person, the same thing's going to happen if they take the same product. There's a lot of commercial variability in the market. You've heard a little bit about that already. Conditions of use might vary, composition of product, and the individual patients, even if they look the same, often aren't the same. So this, again, makes it hard for you to get that tight linkage. Pharmacovigilance in general, this is what it basically is. It's a voluntary program, very rare events, so this is the only way you can find them. And there can be static, and this is for your slides so you'll know where you might go to look to report stuff. It can be dynamic. So for example, people report in real time, either in the emergency room as you come in, but there's also new mandated reporting that a new law about 4 or 5 years ago put into place, reporting to the FDA for any reported side effects of dietary supplements and over-the-counter medications. So that's been quite helpful, and I'll talk to you a little bit about that kind of data in a second. So post-market surveillance can be either disorganized or non-existent or informal, or it can be quite an organized process. And in Europe, it is an organized process for dietary supplements there. You have to do a formal post-market surveillance study. This is nice because it again gets into the real world and to patients doing what patients do, and it gets you to populations you might not normally see in a clinical trial. And it gives you kind of a snapshot of what the real world taking of this medication might look like. Okay, this is the new legislation in case you really want to take down that number and read it. But what it basically is it's mandated reporting through the same MedWatch service as everything else. And having looked at that data so far, we're trying to do a series of look for trends over a number of years. Looked at the first 2 years of data. We're trying to get FOIAs for the last 3 years. Anyway, so just in general, there weren't that many events reported, even with mandated reporting. Given the millions and millions of doses, the reporting levels were relatively small. So one thing we'll see is, as this law has gone into effect, have reporting numbers increased as people got more used to this. And again, herbal products were not proportionally reported more frequently. Herbal products were a little bit picking on because they're complicated and they're more likely to potentially be adulterated or some other kind of a problem. People are reporting, but voluntary reports still represent a significant number of adverse event reports. So mandated reporting is not capturing everything that's being reported. We did see one signal, which was also acted on by the FDA, and it was a hydroxycut product. So that one was taken out and was found. So for signal identification, is this system accurate? Yes. Is it necessary? Maybe. And that's another thing we'd like to look at trending over time. Were there events that either didn't get pulled out or got pulled out late? And what do you think the most common adverse event reported in this population was? What? Nope, trouble swallowing the pill. Okay. Now, as we start to look at taking this stuff onto analysis, okay, now we've told you all the ways in which you can find this material, what you might be looking at, you're pre-thinking ahead about causality. So basically, you're not going to really find, again, I said you're not going to find causality. You're mostly going to be looking for sentinel events. And these are events which, after you analyze it, given all the data that you have, you can't find any other explanation. Either the population is very young to have a heart attack, or they had a seizure with no apparent seizure history, no history of trauma, etc. So that's a sentinel event, something you can't explain that makes you go, uh-oh. And then a signal means that you get a number of these. Now, signals usually require action, or they often require action, but it's not like a research action where you're then going to go do a randomized clinical trial to confirm this signal. No, you get a public health intervention where someone decides to send a warning letter, withdraw the material, make a public health service announcement. So sentinel events plus leading to signals lead to other kinds of events. Okay, I'm going to skip this. Now I'm just going to walk you quickly through the Ephedra example of this. This as an exemplar so you can see how these different risk assessments and how the safety assessment played out. Okay, so first of all, starting with Ephedra traditional knowledge. How many of you know anything about traditional Chinese medicine? All right, so you'll kind of get the lingo here, but most people are going to find this like, what are you talking about? So it's characterized as an exterior releasing herb recommended for short-term use only. So I don't know what an external releasing herb is if I'm not in traditional Chinese medicine, but I sure get this part, short-term use only. So people who already, I can tell you from traditional knowledge, it would be against traditional knowledge to use this herb for a long time. Then here's the other thing that it can cause or it can relieve. And how do they use it? Okay, and if you have an overdose, they say it'll take a couple of hours. It should show up a couple of hours after ingestion and resolve about the same amount of time, roughly. So that's from the traditional knowledge. And then here's the pharmacokinetics, so we know what the area of the curve looks like, when the time to Tmax is, et cetera. So you kind of got a sense now of within what time frame of exposure would we be looking at. So this is telling us, this is another one of those things where we show you millions of things and you tell you what you actually get down to. So it turns out that we got 3 batches of FDA files from MedWatch. There were 400 of those, which we did detailed review on. We rejected 87, and these last 333 we did extensive analysis on. Now, because this was a material that had a lot of public interest, there were patient charts attached, which sounds benign, until you realize this patient probably spent 3 days or 4 days or 5 days in the ICU. Some of the charts were like hundreds of pages. So this analysis was very, very rigorous. We wanted to be shown, if possible, what was the exact time the person took the material. Do we have the material to recover? If there was a death involved, do we have the autopsy report? Was there evidence in serum at any point pre or post-death for a measurable level of the ephedra alkaloids in the blood, et cetera. So we were, again, trying to keep pushing this linkage as tightly as we possibly could. So here's what we came up with. Now, this was also, we looked at a whole bunch of randomized controlled trials as well as adverse events. So our charge had 2 parts, one to look at efficacy and one to look at safety. So we looked at the safety from the adverse events reported in the clinical trials. And as you can see, we had some adverse events that were reported a lot more often in the Verum group, in the group that took the ephedra, than not. So this was anxiety, nervousness, irritability. This could be, this got classified as a lot of different things. Heart palpitations, but then tachycardia wasn't really perceived. So some of this wasn't all that consistent, and we could have expected hypertension based on the pharmacology, but in fact, even though this was in a number of trials and had a fair number of exposed patients, it really wasn't seen. So, or it may not have been seen, this may not have been big enough, because you can see there's a difference in the intervention, but it didn't arrive at statistical significance. So it may be that if we had another 5 trials and double the amount of patients, this might become significant. But in any case, some of the things that you expect people to report, twitchy, irritable, nervous, insomnia, etc., did show up as for adverse events. Now, this is the page from our report, which I can tell you how to find that. It's a JAMA article. And if you want all the gory details, I can tell you how to find the big report. This is going to go down by each major adverse event, and we box them into these different boxes. So death we had standalone. Now this is going to include cardiovascular abnormalities that led to death. That got classified as death, not as a myocardial infarction. So this is if you survived your cardiac event. This is if you didn't. Any kind of cerebrovascular accident, seizures, stroke. I mean, seizures were separate, but any kind of stroke or TIA or anything like that, psychiatric symptoms, that we ended up doing as a separate analysis. But you can see here that now you go down and you're going to look at how many sentinel events did we find. So we found, and we also looked at ephedrine in the over-the-counter reports because we thought, just to kind of put this into context, if we're going to look at ephedra, the alkaloid is ephedrine, and so let's look and see what the over-the-counter, because this is contained in over-the-counter products as well. And so you can see that we only identified 2 out of the 11 were sentinel events for ephedra. Now, these other 9 either meant that there was another causal event, or there was another causal circumstance, or some other thing that we thought really could have explained this death. I'll give you one example. And we had a big fight over this. So there was a young guy who wanted to make his weight class, and I'm going to, this is the end of this, and then I'll be able to take questions. And he was put on a sweatsuit, put on a plastic suit, went into the sauna, took his exercise bike, hadn't been eating, took ephedra, and then exercised in the sauna and died of heat prostration. Well, did the ephedra cause that? Or was it one of a series of negative adverse events? So you can see that that's what goes into actually arriving at these sentinel events. If you're very, very rigorous, it can be a very challenging thing to do. So with that, I'm available to answer questions, and thank you for your attention. That's awfully nice to see. Thank you. Okay, I'm sure somebody, yes.
Yes. Thank you. Interesting talk. This is a thing I was wondering about yesterday after some of the talks, and again today. What do you think of clinicaltrials.gov registration for almost any dietary supplements study, NIH-sponsored, industry-sponsored,
Yeah, I think
backyard picnic-sponsored?
Well,
would that be useful for?
I think it would be extraordinarily useful to put some of the more formalized trials onto that registry, just so that we know where to go look for data or who to ask, because we actually, for this analysis, we actually tried to find unpublished trials that might have been held by manufacturers. We, wrote letters, put notices, and so just to know where to go find those trials later, that would be a terrifically useful event. Now, some of these trials, I think you're going to get loaded down with a bunch of stuff that's just not even realistic. But I also would like to see clinicaltrials.gov start to include a section for, is safety data collected? Are we going to ask these questions? Because I think, again, if you want to go back later and look and see, was there a problem with some of these things? Again, it would just be great to know if that was, and it would make people think about doing it more. So.
Thank you.
You're welcome. I enjoyed your talk yesterday, by the way, too. Such a quiet group. Are you really ready for lunch? Is that the problem?
Not me.
Not you. Excellent.
ready.
Thanks, Joanne. I can always depend on you. Thank you so much.
I have 2 questions.
Okay.
The first is on ephedra. It seems to me it was an interesting example of the data that you've shown and
Sure.
did such a great report on. But then the personality factors, and I don't know if this is true or not, but one is that a very high official at FDA's niece, was it niece, Joe? Nephew, he had an adverse event, and I guess he died? Yeah.
Well, all my files were de-identified, so I can't comment about
okay.
that.
And the second was there was a baseball player, was
Yes,
it, over
yes,
in
yes.
Maryland?
Yeah.
And
Yeah.
sometimes the
And you know-
politics works.
Well,
in
you
a
know how
direction-
how they decided that he used it? They found it in his locker, which is true, but I have a lot of stuff in my locker that I don't use. So, yeah, those are the adverse events that publish in the great journal that we're all used to, the Boston Globe, the New York Times, the Washington Post.
Democracy dies in
Yeah,
darkness.
and we never saw that adverse event, actually. We never saw that
Okay.
one.
The second thing is, on TCM,
Uh-huh.
again, I've heard, and I don't know if it's true, but hearsay is common in Washington,
Mm-hmm.
that the traditional Chinese medicine standards for products sold in China are higher than they are for export. Is that true?
I have no way to know that. I know that there's a commission or a Bureau of Traditional Chinese Medicine, and we do know that when you mess up the food chain or the supplement chain, results your punishment. You don't just withdraw your papers. It can be quite harmful. The group the minister that was in charge of the group that passed the melamine milk and protein, he was actually executed. Yeah, so happily that doesn't happen here. Yes.
That was a nice talk. As I'm guilty of doing these larger trials and thinking about when you're
I was
conducting.
thinking about you.
Well, but when you're conducting these types of trials, no matter how small, medium, large they are, though, I mean, how optimally, when you're doing trials of dietary supplements, should you be infusing all the safety, adverse event reporting, and all that too? Because, like in a larger scale trial that we do, it's harder to dive into the type of detail that you can with Ephedra.
Right. So you're not going to potentially sit down with someone and have an interview and first let them tell you, ask an open-ended question, did anything unusual happen in the last whatever? And then follow on to the ones that you, just by knowing the pharmacology, the physiology, you think might be more attributable. But like I said, I think there's an adverse effect of doing too detailed an intervention. You lead your subject, and I'm a perfect example of that. So what I would say is you ought to make it very, very easy to have people report back to you anything that they didn't notice, that they noticed that was unusual. Now, if you have an observant group like a physician, they may tell you my urine turned yellow. Well, but they're taking a B vitamin, so some things are just going to be a wash. I think also it's not unreasonable to include some basic safety data, and even if that's not you doing the test, but it may be just getting permission signed to get laboratory values from their physician of record, et cetera, I would really recommend that. So, so I think just being creative and thinking about this, I suspect that you could actually end up with some really valuable data. Again, you're randomizing so many people that I think it would be tremendously useful. Duffy?
Yes, Dr. Hardy, thank you. So you did a tremendous job sort of depicting the difficulty of establishing causality when you drill down and get the patient files, you do all of that.
Right.
What do you think of people who are using things like emergency room visits and data from the Center for Disease Control to try to make similar statements about the safety?
There's the Dawn Network, there's emergency room visits, there's a lot of different ways. The thing is, there's a lot of ways to access this kind of spontaneous occurring events kind of problems, and at the end of the day, no matter what you're shown in that kind of data, it cannot attribute causality. So we presented this data that we'd done on Ephedra, which was clearly the longest, deepest. It took us a year. We had a whole team. It was done at the Southern California Evidence-Based Research Center at the RAND Corporation for the AHRQ. There's no one that's going to redo that kind of a study. It was enormous. Even at that, when we presented to the Health and Human Services group, they were like, good, you proved it, we're done. And you remember there had been a prior judge's ruling which was reversed because causality was inferred when it couldn't be proven. And we said, yeah, we can probably prove this. It's going to be a case-controlled study. It's going to require 10,000 participants. It's going to cost X millions of dollars, whatever it was going to cost. And at the end of the day, that was not feasible. So a public health decision was made, which is a whole different kettle of fish, and people should be clear to draw that distinction. And I have to say, FDA went way above board to investigate these serious adverse events. A lot more data was obtained for those adverse events than you have available for, let's say, your average adverse event report. So you know, I think as long as we don't feel like we can do causality from that, we can raise attention, raise alertness. Certainly, if any of these materials are put into a clinical trial, additional safety data should be collected. Continue pharmacovigilance to the point of finding a public health risk, and that's what happened with Ephedra. So, thank you guys.